Hello everyone and welcome back. This is the second systems biology practice session and we will be talking about protein associations. Usually, in a cellular environment, a protein does not work alone. Therefore, in order to understand a protein's function, we need to know the proteins that are associated with it. It can be a direct association, such as physical binding, or indirect, such as a protein that are part of the same metabolic pathway. Today, we are going to see the string database that provides data and graphical representation of protein associations. We will take the IDH1 protein we had seen in our previous practice session as a part of the citrate cycle. Let's enter the string database website. On the main page, we will press Search. There are several options for protein search, and we will search our protein by its name. We can also specify the origin organism. I will not specify that, and let's see what happens. Since I chose auto-detect for the organism, I can choose whatever organism I want from those that express our protein. I will stay with Homo sapiens and press continue. We can now see the protein's string network. Let's review the results page. As described here, each node represents a protein. We can see our query protein, IDH1, with 10 highest ranking proteins, its closest associations. The colored nodes are the first shell of interactors, which means proteins that are directly associated with IDH1. Our query protein is colored in red, right here. Proteins from the second shell of interactors that do not appear here yet, that is, proteins associated to these 10 proteins but not directly to the query protein, will appear in white when we'll choose to add them to the network. The drawing inside the nodes is the protein 3D structure, if exists. What do the edges represent? There are 3 options for the edges, and they can be changed on the settings menu. In the default option, Evidence, the edges represent the protein-protein associations. We will elaborate later on these associations. The edges can also indicate confidence. The thicker the line is, the stronger are the data supported. Another option is that the edges show the molecular action, whether a protein has a positive or negative effect on the other. But let's go back to the evidence view. There are 8 types of associations. Each has a different color as specified on the Legend tab. And they are seen here. If we scroll down to the table of associations, we can see the strength of each association between the query protein and each results protein. The darker the dot is, the stronger is the association. If we stand with the cursor on the dot, we will see the score for this association. Each protein also has a combined score, shown at the end of the line. Clicking on each association dot will reveal more information regarding this association. Conserved neighborhood show genes that occur repeatedly in close neighborhood in prokaryotic genomes. This may indicate a shared function or being part of the same process. Clicking on the dot will show us different prokaryotic genomes and the appearances of the query and result genes. There can also be gene fusion events in other genomes that can indicate shared function, and they are shown here. In this case, our protein has no documented gene fusion data. The occurrence view shows the presence or absence of linked protein across species. If both genes appear together in other genomes but not alone, it is a strong association. As seen here, we can see instances where both genes appear in the genome or do not appear together. Situations where only one of them appear in specific genomes, are very rare. The co-expression view shows the genes that are co-expressed together. In experiments, we can see a list of significant protein interactions gathered from other protein-protein interaction databases. This is a physical evidence that the proteins interact. In this case, the Homo sapiens information is taken from the BioGrid database of protein interactions, and we can see that the IDH1 and the IDH3B proteins have a physical interactions that was detected in a specific assay, and this interaction was also found in other organisms. The databases section shows list of significant protein interaction groups and pathways that both protein appear in, gathered from curated databases. In this case, the third result is the citrate cycle we had seen before. Text mining shows papers that contain both proteins on their title or abstract. The papers are taken from PubMed, as seen here. The last one is homology. It does not elaborate on the score, but it means that a homolog protein of our query protein is associated with this results protein. These associations are both direct and indirect, and can help us to better understand the relationship between our research protein and other proteins. In the Setting tab, we can change the view parameters. For example, we can change the minimum required interaction score, or the number of interactors to show from the first or the second shells of interactors. We can also export this data to different file types, as shown on the Exports tab. We can also do advanced analysis of our network. Pressing on the Analysis tab, we can see whether there are enriched Go terms for each aspect: biological process, molecular function, or cellular component, for all the proteins in our network. We can see publications that contain several proteins from our network, how many proteins are part of the same pathway from two databases, KEGG pathways and Reactome pathways. We can see keywords from UniProt that are shared between our proteins, and see whether there are proteins domains that appear in several members from our network. The protein domain databases are PFAM, Interpro, and SMART. If we think from a perspective of drug development, this analysis page is very important. The network itself can help us understand more about the protein and its function. The analysis page can also help you with that and with the protein cellular location, but moreover, it can relate you to relevant pathways that can be targeted. If we look again at the KEGG section, here, the first result is the citrate cycle that we reviewed in our previous practice session. Of the 30 genes that are part of this pathway, 8 are in our string network, and as the current network contains 11 proteins, this is a substantial fraction. If we press on the pathway name, the proteins that are part of it are colored in red. We can see here this group of proteins. If we press on the ID on the left of the pathway, here, the KEGG map page will open. From this page, we can go to the pathway entry page, here on the top, and learn about the pathway. We can also review related diseases, as we've seen on our previous practice session. Today, we've seen a way to generate a network of associations for a given protein, and how to assess these interactions in order to better understand our protein and its activity, as a first step towards drug design and discovery. Next time, we will see a database that contains information about existing drugs, and we will see a possible approach to drug design.